So it's a real pleasure being here. I'm so excited to be at this conference. Yesterday, the talks were wonderful. The poster session was really exciting. So it's really a pleasure to be here. Thanks to the organizers. So my work is about the mathematical microscope, as I call it. This is to look at viral particles through the lens of groups, graphs, and tilings. And I hope I can convince you by the end of this talk that by doing so, we can make discoveries in biology that would otherwise not have been possible. Of course, they are jointly with experimentalists. We're working very much in tandem with experimental groups. But as you will see, the mathematics always gives an angle of novelty that allows us to find needles in a haystack, if you wish. And for me, the story is really interesting from the point of view that we now understand mechanisms of infection and of viral evolution so well that we can actually turn tables and exploit those insights in collaboration with industry for antiviral therapy and gene therapy. So it's very much a story that goes in both directions, as we see on this poster here, math-inspiring biology and biology inspiring math, and I hope to end up at the end of my talk with showing you how the mathematics we have developed with inspiration from the biology actually is relevant also in chemistry and other areas of science. So hopefully spreading the whole breadth of it. So, symmetry in virology. If you look at this zoo of viruses on the left-hand side here, what you see is that they all have one feature in common, and this is their symmetry. Symmetry for me means a specific arrangement of symmetry axes. You see them here depicted with reference to an icosahedral frame. These are 5-fold, 2-fold, and 3-fold symmetry axes. And that's what drew me initially into this topic. But what you also see here is that viruses are very distinct. So the question is, are there other mathematical techniques we can develop to actually better understand this variety in viruses? Now, you might ask, why do viruses exhibit symmetry in the first place? And this is due to the principle of genetic economy, which is to say that viruses obviously need to code for their components, which costs capacity in terms of the length of the genome. But if a virus codes for identical building blocks and reuses them many times to build these containers from multiple copies, then it achieves a relatively short genetic sequence with a relatively large volume to encapsulate the genome. And indeed, this is what this principle is all about. But as a mathematician, then, you ask the question, why icosahedral symmetry? And this is really a group theoretical question. This is to ask, what is the largest symmetry group you can have in 3 dimensions? And that is the icosahedral group. So we can get the largest repeat of our base unit for this assembly of a viral particle if we use icosahedral symmetry, and this is what nature hit upon during evolution of viral particles. Now, as I said earlier, we need to understand beyond the icosahedral symmetry how viruses are constructed, and as we say, the size of the group, so after the icosahedral group is 60, we have 60 elements in the group. That means we could only have containers from 60 proteins if it was strict icosahedral symmetry. But we have much larger viruses. You've seen a lot of examples also recently in the news. So how can we understand that variety? And the first results along those lines were by Kasper and Klub. This is classical results by now. This is a theory that is to this date used to classify viruses in virology. So this is a very simple mathematical idea here, and we will see during this talk how we need to expand on that to understand larger groups of viruses. But it's a very nice first example to show you how viruses really are constructed. So imagine you have a lattice, you have a grid here. So in the first talk yesterday, we saw lattices very much, so I'm excited about lattices. If you have a lattice here, and they ask the question, can I wrap this lattice around this icosahedral shape in a way that you have no gaps or overlaps? And indeed, you can do this and you can classify this. And this is the work by Kasper and Klug and led to this series. The T number, as it's called, tells me how many small triangular faces I have per large face of an icosahedron. That theory was around for a long time, but then when you look at applications in nanotechnology and look at this group of viruses here, these are plant viruses, small viruses, you see they are distinct, but in Kasper-Klug theory, they would all be the same. And clearly, they're not working the same. So there is something else we need to discover here to really fully understand what's going on. So again, we are wanting to focus on those viruses at the beginning of the talk, which are all part of that same element of the classification. But let's see if we can do a bit more to tease out what these viruses really are about. So in order to do so, we have to think about the relation between the math and the biology. Why is a hexagonal lattice actually a good tool to describe viruses? This is the case because we use identical protein units to build the capsid. So the local interactions between them that are important for the formation of the capsid will be the same throughout the container surface. So from a mathematical point of view, we are thinking about a tessellation that is vertex transitive. So the vertices are where the proteins are meeting, and so we want all of those interactions to look the same. But if you think about what other lattices you may have that are vertex transitive, then you see the classical Archimedean lattices shown here on the left-hand side, and all of them have that property too. So you can ask the question, are any of the others actually also fit for the same construction? And that is a paper you will see on the next slide, but I want to first show you here one virus, a herpes simplex virus, that actually follows one of those other lattices. You can clearly see there are the hexagons, but interspersed you have these triangles as a Kagome lattice. So clearly other lattice types are important. So what we did here with Tony Luque at that time, it was a Nature Comms article here, where we looked at these Archimedean lattices and we asked the question, can we redo that construction, that classical construction? And what we found was actually quite exciting from a biological point of view, because in between those geometries from the earlier classification, we found others that had sometimes the same numbers of protein or different numbers of proteins, but they were sort of stepping stones on an evolutionary scale between the sizes of the classical architectures that were known and characterized. And as I'm going to show you at the beginning of the talk, this really allowed us to drill into the biophysical properties of those viruses and make us understand better how they work and which ones we should use for which applications in nanotechnology. So let's dive into that. Let's also mention that these other lattice architectures have new degrees of freedom that are very important. So here see again this lattice that is a mix of hexagons and triangles. And as you can see, you can vary the edge lengths of these different polygons in this lattice. And that is actually a degree of freedom that viruses exploit. Small viruses, large viruses. So you need that wider mathematical framework to really understand this. Okay, so let's go from structure to function. We understand that we can use lattices to model viruses, but let's see what it really tells us about biology. And in order to do so, we are looking here at these three examples that we saw on this slide with the nanotech applications. So these are three viruses that are formed from the same number of proteins. It's always 180 proteins, a T equals 3 structure. But as you can see superimposed, the way in which the proteins arrange themselves are different and can actually be described in terms of a triangulation. In this case, there are 3 proteins. The colors represent individual proteins. So you have here a triangle with 3 proteins. Here you have a kite shape. I'm not sure if you can see it. There is blue, green, and red making a kite. I point a kite out for you. And this is a rhomb shape. So you have 2 types of rhombs in this picture. So 3 different types of tilings. And what we have at the bottom are the interaction networks. This is to say, put a vertex, the dual of the tiling, so put a vertex into the center of a shape and connect vertices if there's an edge between them. So this is obviously important to capture the interactions that these building blocks can do. And now I invite you to do a very simple experiment. Again, we can do this more complex later, but just from this simple experiment, we learn a lot. So let's think about these three scenarios again. So the ones with the triangle and the kite each have 60 units because each unit has 3 proteins, but we have 90 units for the ROM tiling. Then if you look at the connectivity of your interaction network, 2 of them share 4 coordinated networks and 1 is a 3 coordinated network here. And now we do a very, very simple experiment computationally. Imagine you have these tilings and you randomly pick out units and you ask the question, at what point does my viral shell decompose in 2 disconnected units, not obviously counting what you've taken out. You're counting the rest, the bulk of your capsid. So then what we show here are these plots where you have the fractional vertices removed. I have to do fractional vertices because there are different numbers of vertices, different numbers of plaquettes. So in order to make it comparable, we take the fraction I've removed. And on the y-axis, I show you how many disconnected components I have. So then I do this simulation 1,000 times, and I see what I'm getting. And the dashed lines are showing for me the moment where for different scenarios, we have basically these 2 disconnected components. And what we find is that this happens at different fractions of removed vertices. So this is to say that these lattices have different stability properties, and that's very important for their function. So the triangular tiling falls most readily apart. The most stable is our kite tiling, and the ROM tiling is in between. So let's see what this tells us. So first, the question is this something viruses can actually exploit more widely? And as I mentioned previously, there are more complex architectures. Could they actually have something to do with stability? So here is a virus that is a cancer-causing virus. It's a polyoma virus, a papilloma virus. What you see here is that these viruses have all over their capsid groups of 5. And after what I told you at the beginning, obviously we can't describe this with lattices wrapped around an icosahedron anymore because there are no lattices with local 5-fold plaquettes. You can have, however, the closest thing to that, which is the example here of a Penrose tiling, which are aperiodic tilings that basically have that symmetry of 5 or 10, but they are infinitely extended, but they have no repeat unit to form the lattice. But nevertheless, in a compact setting, you can use them. So I put a lot of mathematics on one slide here. So basically, if you want to construct these lattices, what you do is you embed your group into a high-dimensional space where the non-crystallographic symmetry becomes crystallographic, which means there is a lattice, in the proper sense of a lattice. We have a unit that repeats periodically. That builds up my lattice, and the lattice shows the symmetry that in a lower dimensional space is non-crystallographic. So there are these cut and project techniques. Again, I'm available later. If anybody has interest in any specific thing from my talk, I'm happy to answer all your questions later, but I want to cover a bit of ground in this talk. So I'm just giving you the overview of a couple of things here. But what you can do, you can repeat that construction that you can do to get a Penrose tiling, to get 3-dimensional tilings that are aperiodic. So you can then view your virus as a section, as a spherical section, you see it here, in a tiling that in this case is formed by these units that have by constructions the correct angles for the non-crystallographic symmetry. So we're ending up with a tessellation then that is in terms of rhombs and kites, very much like what you've seen in the case of the Penrose tiling. And it would just be wallpaper, meaningless wallpaper, unless there is a one-to-one mapping onto the biology, which there is. So the way we interpret this is that protein units are sitting in the corners of those tiles that have this 2^5 angle, so obviously not here. In the 2^5, it's like the kites we saw earlier on the screens. Or in the ROMs, they are sitting on these opposite edges. And then with this in mind, you can one-to-one make a mapping between your virus and your tessellation here. Now the question is, can we do that same experiment again and we find something really interesting? So imagine we have this tiling now, but my units actually are the pentagons and the hexagons. So here I superimpose for you the pentagons, and the hexagon, as you see, has a 5-coordinated vertex. It also harbors a pentagon actually because of the 5-coordinatedness. Remember, we have a protein at vertices of 2^5, so you have 1, 2, 3, 4, 5 in this hexagon as well. So this is another way of representing this, but the interactions between those proteins are not the same across the capsid. Remember, we have these ROM shapes where we have interactions between 2 proteins, or we have the kite shapes where we have interaction between 3 proteins, like a daisy chain interaction. So this means when we are looking at the edges across which we have interactions, we need to give them labels. They have different labels. We have A, B, and C, depending on how many arms and how many types of interactions there are. So we have 3 types of interactions, but on one we know it's 2 arms that are identical. I know it's a 2A, but B and C, I don't know, right? So I have 3 parameters now. So now let's see what our virus does with those parameters. So now we do the same experiments. Take your plaquettes out. So now we talk, it's a little hitch here, we talk about energy removed. The energy is simply we are looking at the fully formed container and we sum up all the bond energies because now we have 3 parameters. The easiest to take care of that is basically to give them labels and describe the energy as the total. So then we are normalizing away one of them, so to have just 2 parameters and make a surface over that. And then what we find is really, really interesting. The virus actually finds a very, very stable situation in this case. It can tune, it has these extra parameters to tune its stability compared to a Caspar-Klug situation, which would be the hexagons and pentagons that we would otherwise get unless we have this special geometry. So this geometry is actually an opportunity for a virus to tune itself to its environment and to what it needs to do. So, and that's something we need to know because we want to do nanotechnology. We want to use these containers to transport genetic cargoes for gene therapy. We want to do drug delivery. We really need to understand how they form, how they disassemble, what their properties are. So I invite you here to, this is a paper we had in Science with a group, an experimentalist group, a nanotech group in Switzerland, where basically they use not a viral protein, but another protein. It's an enzyme, as we say, that is forming dodecahedral cages, so the cousin, the dual of the icosahedron, if left alone. But it's not being left alone, it's actually engineered. So what our colleagues are doing, they are fusing the protein, the ends of the amino acid chain, and then they cut it somewhere else. And the reason why they do it is because they want this particle to recognize its coding sequence as messenger RNA. Yeah. So then they take the protein and they subject it to rounds of evolution in a laboratory environment. So we can literally look and see the evolution of structure in a laboratory environment here. And what you see is you gradually get larger and larger particles, and you get these particles with these interesting holes here that become larger and larger. So there is this, I should say, there is this selective pressure to encapsulate. So after every round of evolution, we have a way in the lab to digest away anything that's not encapsulated as RNases. These are enzymes that eat up anything that's not protected on the level of RNA here. So, and we obviously trace the start. That's the selective pressure. But then we see how this is evolving, and we want to understand this. So now let's look at those particles that we're getting here. We want to understand their properties again. So interestingly enough, this is here. One of them is again a particle that is formed from 72 pentagonal shapes. We call them pentamers. Exactly like the virus particle we looked at with the tiling earlier, but it doesn't follow that tiling. It has these holes that viruses usually don't present, but these nanoparticles can. But on the right-hand side, you have again a particle that is a very classic structure. It's not quite a Caspar-Klug tiling. It's one of these Kagome lattices, but it has 12 pentagonal shapes and 60 hexagonal shapes. Same number, different geometry. And now we do our experiment again. And what do we find? Very interesting. We look for two scenarios. Hole formation. So this is to say, when we do our experiment, we take plaquettes out. We record if a hole of a certain size is formed. Then we say hole formation has taken place. Or it might fall apart before holes form. So we just take out and we see what happens. Do we get a big hole of a certain size? Do we have two components? And you see this here in this, so the fragmentation again, this is the thing we did before when it falls into two parts. And very, very interesting, your particle, despite its holes, the one with the pentagonal shapes, despite its holes, it's much more stable. You see the red dashed line is behind the blue dashed line. Whereas when it comes to hole formation, this is actually happening later, but the order is much closer here. But what you see is clearly that your particle, is called ALS, is the nanoparticle, is much more stable. And indeed, this is a nanoparticle that our collaborators now use for nanotech applications, and we are working together. That was, when we started working, the lady who was the postdoc at that time, she's now running her own lab in Lausanne. And I've just seen her 2 weeks ago. We're basically continuing this. We're using those particles for nanotech applications right now because of their properties, because actually they are really good for us. Also, we learn a lot about biology. We've seen this other container here, and we've seen it's much less stable than the container that we've gotten from this evolutionary experiment. And nature actually therefore has a problem. It has to stabilize this. But the way it does that, it puts an extra protein around, we call it a chainmail organization, to provide additional stability around the container. So this is something that we now understand why that is, because simply the stability properties of that particle aren't enough for the virus unless it evolves an extra component here to take care of that. So really what we are doing is we're using these protein-engineered capsids for our applications. Now, because we do so, we really need to understand the mass. And by that basically that work with that group in Lausanne has really prompted us to rethink the whole mathematical framework of these lattice arrangements. So clearly, just to recap, we have seen these typical surface tiling in the Caspar-Klug theory. You can think of triangulations or hexagonal lattices. They're dual of each other. So this is pretty much this classical theory. Then I showed you that then we can actually think about other Archimedean lattices like these types of romp lattices or combination. I also showed you that we can have these arrangements that are based more along the lines of the mathematical techniques you use to describe aperiodic tilings. However, now what we, and that was our PNAS paper with my postdoc Farzad, that we had here, the question is, you can't really describe this very much as surface tessellations anymore. The reason being is, as I said earlier, tiling doesn't mean anything unless there's a one-to-one correspondence between units and the structure, because otherwise you have a nice, beautiful wallpaper, but no insight biologically whatsoever. So that certainly didn't work. So what we then did here is to develop a new theory of interaction networks. So here, what we're doing in a nutshell, we completely strip away the geometry of the individual building blocks. So the fact that these are pentagons or hexagons, we don't care. We give them a dot, a point, and then we say we connect them if there is an interaction between units. So what you see here are pretty much tilings that describe interaction networks. Now, these tilings have different properties. If you look at the right-hand side first, there is no more reason that they should be vertex transitive, because you can have these different types of arrangement if it's not a lattice. If it's a lattice, fully covered, no, but this has holes, so yes, we can. So what do you do mathematically then? Well, you see the local interactions, and you can see the local interactions here are what we call a squashed hexagon. For want of a better word, it's a square with two triangles, yeah? And so you ask the question, can I classify all possible connectivity tilings that have that property? So what we did in this work is then mathematically to enumerate all possibilities and then fold those up into symmetric shapes, giving us all these types of particle shapes that have, these 4 of them have indeed been discovered experimentally, but we also predict 2 more that have not yet been seen. So we cover everything that has been seen plus extras. Now, why are we excited about that? Is this again wallpaper or is it useful? So we want to be developing mathematics at this point that is really giving an impact in the biology. And I give you here an example of why this is predictive. We want to be predictive, yeah? So this is a completely different protein. It has sort of a globular body that forms 3-fold axes with a linker that forms 2-fold axes here. And what you see here is a particle, imagine that is one of our particles, that we decode mathematically by basically deciding we convert it into our interaction network by putting a dot at the position of a protein, and we are putting a link when 2 proteins are together. And then we can unfold this, we can, if you wish, embed it in the plane to get this lattice. And now what our theory says, any other embedding of a symmetry, so that can be a square, that can be an icosahedron, that can be tetrahedron, right? That is such that when you close it up, it's without gaps and overlaps, gives us another solution. I think there's a rogue pointer here around. If this rogue pointer could stop, would be lovely. Yeah, so then you have another embedding here. And you see this different embedding, when you close it back up, you get another structure. And that was then subsequently seen experimentally. So what this allows you to do is, once you have one structure here, then you can decode its building principles with this, and then use that understanding to create other structures you may not know about, but that you could use in your applications. And I have here a couple of papers, recent papers, where we use this approach with collaborators internationally. So this here is work with a group in Australia, where we are looking at not viral containers, but these are encapsulines, so other type of protein containers. So this approach enabled us then to construct particles of different types of symmetry. So that's a recent paper. Another recent paper is with a group in Poland, where we used a similar approach to understand tubular structures that are formed from these protein units. So they knew about one of them, but they didn't know about the other, and we were able to help them construct those ones that they then subsequently were able to resolve as well. Another piece of work that we are quite excited about is with the Baker Lab. It's close to home for you guys, Seattle, so University of Washington, of course, with Neil King and David Baker. So what these people are doing, they are designing proteins. They're no longer adapting proteins from nature. They say, we go in the lab and we create our own proteins and we make our own containers. So these are containers that are meant to be constructed from units that form three-fold axes, that have three arms. And when they did so, what they wanted was a particular geometry, a particular container type. But it turned out they got polymorphic assemblies. They got a whole zoo, you see it down here, of different particles. So they came to me and said, Reidun, that's your expertise. Can you help us sort of understand what we're getting and why we're getting certain multiplicities of some of those and not of others? So then what we did here, and this is maybe a nice example where geometry is being braided with other techniques. So what you do here is you do MD simulations of your building block. And the building block has different angles between the three arms with a certain distribution. So you see here the angle distribution that comes from an MD simulation of your building block. So it tells you where your building block is most happy at the peak and where you stretch it a bit too much to get the angles. So then you can do a full classification of anything that's at all mathematically possible. Then you can record for your discrete choices, discrete options, what the angles are that are needed to make that polyhedron in these places. And then we introduced this concept of a minimal frequency of angles. So I talk you through. Here is one of those particles. Then you have these different vertices. At a vertex sits one of my building blocks, and it has certain angles that it has to take on to fulfill the role of that vertex. Now, by symmetry, a lot of them are the same. In fact, we have only three distinct options in this case. And if you now look at the angles and you go back to your MD simulation, you can see that some of them are at higher frequency in the ensemble and some are at lower frequency. So the minimal frequency is the one that's the least frequent in your ensemble. So this is your weakest link. And what you can then plot for all the options, so on the x-axis, we list all these options by the number of trimers, by the number of vertices or building blocks, as you want to call it here. And on the y-axis, we plot the minimal frequency for each of them. And wherever the minimal frequency is the highest, this is what you would expect to see most readily in your ensemble of different options in the experiment, and that's what happens. Yeah. So from this combination of geometry and MD can then explain what you're seeing, and it helps us now, we are continuing this work, to design, to home in on that knowledge and basically make sure that only angle ranges are possible that give desired outcomes, for example. Okay, so I hope I could show you until now that we have come a long way in understanding the morphologies of these protein containers, and our dream really is that to the designer containers. We are actually very, very much in that now, in that phase. But I have suppressed the other main star of the show, which is the genome. So what we discovered, and I will show you in a minute the mathematical side of this discovery here. This is obviously an interdisciplinary project together with experimentalists, but I'll show you the mathematical side of that discovery in a minute, is that in viral genomes, they are not just letter sequences. These letters can self-interact, they form these secondary structures. Then in these secondary structures, you have these sequence snippets sitting at specific pivotal positions. So in this case, when you have these loops of these stem loops having an A, XXA, XX meaning any nucleotide, just two of them, your choice, yeah, then these are able to act collectively by attracting protein and helping the formation of the container along. Imagine you buy an IKEA shelf and you have the instructions. So here are the instructions of the virus for its container. How do I place myself together, right? So we have a lot of modeling. Talk to me later. I had to make choices for this talk. We have lovely stochastic simulations that show how this mechanism works, give us insights into the relative affinities for protein, how this is tuned evolutionary. There was a lovely poster yesterday on a sequence space versus phenotype space. It's related to that. Talk to me later. I will not be able to cover all that ground, but what I want to do is show you again the mathematical microscope that helped with that discovery here, because I think that's something probably this community would enjoy to see. So, viral geometry informed data analysis. We want to find the needle in a haystack using math. And let's see how this goes. So the challenge mathematically here is we have not only got a protein container, we have packaged genomes inside. So a lattice or a surface lattice lives obviously at a radial distance from the center, but how can I extend? I want to stretch you a little bit more now in a radial direction, not just think about different container options, but can we understand the relation between a container and the packaged genome here a little bit more? So the math is actually surprisingly simple at that level. I'll stretch with a few formulas later, but the main idea is actually quite simple. So when you have a lattice, this means you have a base unit that you can translate in a specific direction by a specific length to get another structure. And if you get it such that the edges touch here, you can imagine how you build up a hexagonal lattice from that by using the rotational symmetries of the hexagon and the translation. Simple so far. Now, when we come to these aperiodic structures, the problem is when we have a non-crystallographic symmetry, we can't do this. Imagine a pentagon. If you try to move it and glue it, you get gaps. You can't help it. So the next best thing we can do is to translate it in a meaningful way. What is meaningful from a group theoretical point of view? It means we are not having the free group. So if you write down your elements in your group, if they have relations, so if you combine two of them and they have the same outcome, if you combine them in a different way, these are relations. So if there are any non-trivial relations, we haven't got the free group, we have an interesting group. Okay, geometrically, if you don't know about groups, don't worry. Geometrically here, this just means we are moving our structure such that when we then rotate around the center again, as we do here to get the green units, we need these so-called coinciding vertices. Whenever I have a vertex that coincides, it means I have different ways to get to that vertex, and I have a group relation. So if you don't know about groups, don't worry about it. Just think about the solutions mathematically of which translations you can use to get these coinciding vertices. And obviously you can already appreciate it's a discrete set. You can write them down, you can classify them via this geometric intuition here. Right, so here is just a little bit more zone out if it's not you, but if it's you, there was someone who said yesterday, blame the man called Lie. I blame the man called Lie because this is related to Lie groups and root systems very much. So I just wanted to bring it here so people know that this is in the background, can ask me later when I'm available for the discussions, if not, just zone out a bit. So these are basically two root vectors here, so we can use what's called root vectors to describe these reflection groups. So in a sense, these are groups that describe reflections and off-center reflections that you get by Kac-Moody formalism. So you extend your root system by another root so that to generate these translations. So ask me, I'm happy to go on a board or on a piece of paper and show you this, how this works if you want to know. But bottom line is, imagine you have this embodiment of your root system, call it a decagon here, and you do the same thing again. You move it, then you rotate around the center, strip away all the edges. What you see here, and this is mathematically not an accident, this is actually intentional, we can chat about this later, you generate vertices of a Penrose tiling here. And if you do it again and again, the next level brings you the red vertices. You get more and more of those vertices, very densely filled at the center, moving out to the outside here. So here's a little movie, and I'm not sure if it's running, but if not, oh, it's running. So this shows you the thing in action, just if you want to see how it works. So we had this little tool here, so you can take in how these affine, this is an affine extended group in 2 dimensions with a non-crystallographic symmetry. It's very visually sort of telling the story as well, so don't worry about the roots if you are not familiar with that, but think about the principle, think about the intuition, the mathematical intuition of it, right? So we're stripping away again, so this is what we get from the first set, and then we would technically now do this again and get a second set, right? So you see how we are really reproducing that. So, okay, so let's go forward because we want to, so now one more stretch. We've done this in 2 dimensions, do the same thing in 3 dimensions. So this group that we've just seen is a subgroup of the group, a reflection group that basically describes or generates the rotational symmetries of the icosahedron that we saw. So just imagine you can embed this. This is the icosahedral group, and I promise you it's only 2 slides and then we're going back into applications. But basically what you here see is the reflection planes, and the root vectors are perpendicular to these reflection planes. So when I think about this process here, I think about this setup, but you can very well just think about the geometry that I described before. It's totally fine. Again, I blame the man called Lie. So for those of you who know Lie groups, basically we can, from D6, we can project this down. So this is what I said about cut and project. We have a regular lattice with the symmetry in 6 dimensions, and it gives me a non-crystallographic symmetry in 3 dimensions. So I like to think in terms of a lattice because I can do integer additions, which is much nicer. Ask me all about this later if that's of interest for you. So affine extensions, you extend again and you get your settings, or think about anywhere I can move my icosahedron to get the non-trivial structures. So I've got again a visual representation of the idea in case that's easier to understand. So this is the root system of that symmetry group. The vectors pointing to those vertices is the root system. Now again, we translate a meaningful way, and we are now looking at that structure after it's rotating around this threefold axis. Here's a threefold axis. Let's rotate around it. And so we generate the other vertices as well. And you see coinciding vertices. That's how it's been constructed, right? So we get our coinciding vertices here. And so you can appreciate how these things build up, perhaps even if the root systems themselves maybe are not something you're familiar with. Now, you have all the tools in a minute that you need to understand how we use this on viruses. So now you can appreciate that these point arrays that I'm going to show you actually represent mathematical objects that come from group theory, that can be classified a bit like in particle physics. I'm a mathematical physicist at heart. So you want to classify particles, or you want to classify viral particles by looking at different options of a symmetry. And here's one of those. And now the question is, what do they tell us about viruses? And that is what's coming up in a minute, because now what we're doing is the outermost vertices are now in pink, and we are going to project them onto viruses. So I had one PhD student who's more on the bioinformatics side, who developed a method, given our discrete set of point arrays, to map them onto a viral shell, so as to find the best possible match. And it's already magic that you find one for every virus. It's not a given. So this is an insect virus, Pariacotovirus, and you see how the outermost features would map onto this virus here. And I've got another video to show us then what the predictions are. I think this is in a different video, which is coming next. Okay. Yeah. So, and here it is. So now we're seeing the next video where we are bringing so this is where we're starting off from the other video. And now we want to know all the other vertices. They come packaged and parcel. They must dictate their relative locations. We only use the outermost vertices to gauge the structure, to find the correct model. But now, does the model tell us anything over and above? Or is it boring? So I hopefully can convince you it's not boring. It's actually telling us a lot. So now we're stripping protein away. You see parts of the genetic material, which is RNA. So in a minute, what's going to happen is that we focus only on 6 proteins here. So these 2 trimers. This is a particle, this is classic, that forms from triangles. We've seen this particle at the beginning of the talk. It was one of the 3 examples from the nanotech, by the way. So now these were the gauge vertices, the ones we used to pick the correct model. Now you see the other ones coming in, and watch those ones here. So in a minute you see how the internal vertices, first of all, it's like an encasing form. There is a crystallographer called Jana who introduced encasing forms. It's almost like an encasing form. But what we're most excited about is now what we're going to see in a minute on the genetic material, which is RNA here. So you see these vertices in the minor grooves of the particle and at the junctions. So that is really meaningful. This is where we actually understand something about how the genome is organized and how the genome, you can see nicely lined up here, how this is interacting with proteins. You get the idea. So how do we use this then? So we have these point arrays, and for a different virus here, you see that these vertices are at the interface of RNA, shown in orange and blue, and protein, shown above here. So as a mathematician, I strip all the proteins in RNA away. I keep only those vertices and I connect them into a polyhedron. And I know that these shapes here, you see them in 3D, which are actually these secondary structure elements, they are connecting at these vertices. So they are actually at these vertices. So for me as a mathematician, finding those cryptic patterns that often have just an AXXA in a loop, hopeless with bio. And if people have tried and they've concluded there are none, and actually this is not true, they are there and they're super essential for the functioning of the virus. But if you play basically the Ecosian game, so if you look for Hamiltonian paths on this polyhedra, suddenly you can unravel the whole mystery and you can pinpoint all these signals. And this is how we helped our experimental collaborators to discover them. So we discovered it first in this virus at that time, and to this date we kept discovering them in major viral families. So recently, last year, I spent 2 months in San Francisco at the Gladstone Institute doing this for coronaviruses. They are there, they're everywhere. So these signals are really everywhere, and it's a big industry, and ask me later if this is something that interests you, but I really have to move forward a bit. So just making sure I'm good. So basically then, so this Hamiltonian path idea led to a novel way of interrogating viral genomes. And think about the Ecosian game. So Hamilton himself introduced it. This is a Schlegel diagram, a dodecahedron put flat on the plane, and people were meant to create Hamiltonian paths, so connecting paths that visit every vertex precisely once. So that same idea of the Ecosian game is basically the crunch point for this data analysis method here that we've been using. So yes, so in a nutshell, you have your potential candidates, but you have way too many because it's not very, an AXXA is very abundant, as you can probably guess. In the genome, you have more than double of what you really need. But then it has to fulfill these Hamiltonian paths organizations. And if you interrogate for that, you will suddenly find that it reduces the ensemble pretty much, and you can actually do this. So, but now, alpha and extended groups, I want to go back from biology inspiring math, because we had a math conference, so ask me about everything else, but I want to use the last time I have to show you how this is actually opening new avenues in mathematics and in other areas of applications. So first of all, we talked about viruses having icosahedral symmetry, but you probably know that DNA has 3 different forms. And the question was, can we actually understand why there are these 3 forms? Why actually we see those and not others? And if you think about the mathematics from a point of view of the root systems, again, of the group theory, yes, you can either take an icosaedral group, but you can also do basically keep a translation with a two-dimensional symmetry group as well. And if you do this and do the same classification, or if you do take the root system of that's underpinning the icosaedral group, you can actually classify all tubular structures you can potentially get. And you will be surprised again how restrictive this is, how few options you truly have. And you get actually the different forms of DNA. So we were quite excited to work with a postdoc, Farzad Fatih. The other thing I really would like to introduce is to show you how much this impacts on other areas as well. So in earlier work, we looked at the buckyball. The buckyball C60 was a Nobel Prize for Kroto here. Is basically a particle which has that same symmetry as the viruses. It has carbon atoms sitting on the vertices of a shell with that symmetry. So it's the famous buckyball. It's exactly 60 carbon atoms. We said the icosaedral group has order 60. There are 60 elements. So give me one vertex and my symmetry group gives you all the other vertices here. And at that time, when we did the multi-shell structures, we looked at what is called carbon onions, where you have these carbon structures nested. And the question is, if you've got one of them, are the other structures constrained? And we answered this in earlier work. We showed that these carbon onions actually follow these affine extended symmetries as well. And recently, I got contacted by a group in China who are looking at silver chloride organizations, where this is just the silver atoms here in, again, nested shell organizations. And with the current postdoc, Kevin O'Brien, in my team, we have been looking at this and tried to understand if our mathematics can also help here. So maybe as a spoiler alert, so what we did then is to look, first of all, if any of our elements in this affine extension can describe these nested structures, and that was the case. But on the way, we discovered something very, very cool. Thank you so much for that. That's brilliant. So we discovered something really cool, and there is a theory in polyhedra which is called near-miss polyhedra. So where for polyhedron, as you probably know, we want the edge lengths to be identical across the polyhedral surface. But then there are people who do near-miss polyhedra, where they say, and this is again inspired by nanotech applications with proteins, where we allow those edges to vary ever so slightly. Now, that was known, but what we find here is a novel near-miss situation in terms of these symmetry groups. So I try to talk you through it on the right-hand side. So again, like before, we had the video, we have a base shape that's being translated and then rotated to give me another shell, like so. And we said earlier, any translation that gives me non-trivial vertices when I rotate is a good one. But here on the right-hand side, if we focus on this, there is a choice. I can either translate so that I get at the center of this square, which is a twofold axis. You can imagine when I then use the symmetry, I get coinciding vertices. But I can also get them at the 5 or at those vertices around the 3. Whatever choice I make, I hit one of those symmetry axes exact, and the others I only hit a little bit. So I have one exact and the others not, so I get a different group relation. And these are what we call near misses, because you see the translation is in the same direction. You see the points that we get with this translation are very close to each other. So the solutions are not very different. And in fact, when you apply to the chemistry, we have done an RMSD to find the correct shell, and we did. But there are two solutions that are very close, and the next solutions are way off. Yeah. So very, very interesting. We have these near-miss situations in an affine extended group, never seen before, for a non-crystallographic group in particular. You would probably not see them in a crystallographic one because you have bigger distances, right? But for the first time, and we're writing this up, so the paper is submitted, but we're writing up the mass story independently of that story as well. Okay, so, and almost done. So what I also wanted to show you, that it has other applications in math as well. So the symmetries here, as we said, give us these tilings, but I can see these tilings now after what we discussed at the end as vertices coming from, in this case, intersecting polyhedra that were moved in space and rotated. And when we strip away those polyhedra, we see these vertices, right? But now you can ask, is it therefore a technique that we can use to understand packings? And it is that, yes. So here we see again an icosahedron moving in space, moving in space, moving in space, creating this kind of extra structure here. Yeah, so it's been moved and rotated around the 3-foot axis. But what if I do this in a way that I'm not intersecting? I say I'm only taking those translations that lead to packings, proper packings here. And we can ask the packing density. What is the packing density for different options? Again, we have a full classification. We have any possible option. For each of them, we can record this. And what we were really, really excited about is to see that here's work from the Klotzer group. I brought this in because we had yesterday a talk from someone who came from Torquato's group, and they also obviously looked at the icosahedral packings in an MD. So I want to bring this in. And you see we have exactly, you see the number that we are getting here. That's the number from a simulation. That's the number we are computing from our affine extended symmetry. So we can get analytically, in this case, at solutions that people find via computational techniques. So it's again giving an extra dimension to the mathematics here. So with this in mind, I would like to summarize and conclude. So I hope I could show you that we use different mathematical techniques in our mathematical microscope. We have discussed tilings, interaction networks, group theory applications that led then to graph theory applications in the context of data science. And the mission that I'm in really is to understand nanoparticles with the aim of having applications. We have industry contracts at the moment. We work internationally with people who have different applications in mind. And it's really an exciting time because you have a special angle through the mathematics that allows you to do things that perhaps would be very difficult otherwise. So for instance, we saw the de novo designed particles from the Baker lab. Then we saw the protein engineered nanocages from the Swiss group. And then finally, virus-like particles. We call particles where we basically take the genome out, virus-like particles. These ones, this is a Picorna virus for work we're doing with Sanofi, our industry partner on this project. So pretty much an overview of the mission of what we're doing here. And finally, I would like to summarize also something I unfortunately didn't have the time to get into, but as you can probably guess, if you understand how a virus assembles, you can find novel ways of interrupting that, is some of the things we're doing. You can create, for vaccine applications, as the work with Sanofi, particles that they need in a certain stability range. You can do that. And finally, in gene therapy, what we're doing is we're stripping away the viral genome and we are packaging in genetic cargoes. And this is another very much important application that I'm very much invested in. Let's put it that way. Ask me if you want to know more. And if you want me not to shut up, ask me about this one because it's one of my favorites. Okay, so with this in mind, I would like to thank my wonderful team. I mean, it's a group effort, as everybody knows. We are working together, especially in interdisciplinary direction. Everybody has their expertise, and we bring all of this to the table and then get at these questions in biology together and our funders that are very generous in the UK. Thank you.
Wonderful talk.
Thank you.
Questions?
So thank you for that beautiful talk.
Thank you.
I was thinking some of the capsid shapes are more common than others, that they can be found across viral families. In your opinion, is there any evolutionary advantage to these? Like, could they be energetically more efficient to assemble or more stable? Or
that's a wonderful question. So when the virus has a small genome, for instance, in small plant virus, it has different challenges because it's easier to form the container. When it gets to what we call a T equals 7 times 60 proteins, it needs scaffold proteins, for instance, to build this up. And we've seen this example where we had this extra chainmail organization. So my answer would be horses for courses. So if it's small, they have not so much variation and you see a lot of the same design. It becomes bigger, they have challenges, and they have different techniques of overcoming the challenges, and you get that variety at that point. And then we're coming to SARS-CoV, where we have membrane. We haven't even talked about membrane-enveloped viruses, where genomes are really large, and then we use the proteins are used to compact the genome and then anchor the genome on the inside of the capsid. Similar mathematics, but a very different design. Yeah,
okay.
Thank you.
Thank you.
Hi, thank you so
Hi.
much.
Pleasure, yeah.
So, I was going to ask about the payload, like how that changes the geometry, but you just answered that wonderfully. So I will comment that there's nothing wrong with making cool wallpapers. And related, are your 3D construction animations available online if I...
Oh, that's Ask Me. So I haven't put them online. Maybe I should. So we obviously have the papers around that, so I could share those. But talk to me. I give them on an individual basis rather than. But I should probably put them online. It's a good idea. Yeah.
Happily, thank you.
No problem.
Hi, that was a really fantastic talk, thank you. I'm curious if you applied your work to any of the more like tube-shaped helical viruses at all?
Yes, very good question. So that's excellent. So that's very similar to the work we've done on DNA. I presented the DNA to show variety, but you're absolutely right. So then you have a translation direction and you have a two-dimensional symmetry and you can do the same idea. Excellent question, yeah.
Really fascinating talk. Have you noticed, is there like so if you're using these for drug delivery purposes, ideally you'd kind of want to minimize an immune response. So have you noticed like any relationship between the geometry and the immune response?
Beautiful question. That's the one that is really important. So the immune response is a real problem for these drug deliveries. So because what people do, say with an AAV vector, right? So they shell out, put the genetic cargo in, but there is an immune response. So when we use this in gene therapy, it's in particular used on small children, where the immune responses can be really strong. And there were in stage 3 trials, where stops being put because the immune response was too high. Now, the problem is that the virus is good at packing its genome, but humans aren't good at doing this at the moment because they don't fully understand the mechanisms, so they try. So at the moment in this industry, people try to purify. It's very expensive to try and purify out all the particles that have mispackaged. And you would be surprised to hear, I was surprised to hear, that only 1% packages properly. The whole rest doesn't. So they need to weed out anything that hasn't packaged to amplify the ones that are packaged well. So not only is this costly, but second, you lose material while you amplify. And even at the end, you have a lot of particles that are not therapeutic. They don't contain the right cargo, but as you say, they're immunogenic, so there is an immune response. So what my trade is to understand what the virus does, mimic that in the cargo, and hence enhance the packaging. That's exactly my application. So by doing so, we are not only pressing the costs down, we are also avoiding those immune response, because if you get the ratio just right, it's just about, okay, you have one pop. So a lot of your genetic diseases, especially in small children, you need one treatment and you're done. It's a one-stop done. But you have to make sure that they basically tolerate that one treatment. And that's exactly our challenge. And at the moment, that's where we're making our big contribution, because we can. So we can do this by understanding what the virus does. We're very excited to talk more about that if you want to know more. Yeah. Okay.
Thank you so much for the great talk. Possibly convoluted question. So when you talk about these near-miss geometries,
Yeah.
you have a fixed point you can reference and talk about how you are missing that fixed point. Do the points that miss the mark, is there a symmetry group about which they are located around the fixed point?
That's a super good question. So let me go back to this picture real quick. If I can go back here, I can. So what you will see in this picture is that what you try to do in order to get a group relation is to hit a symmetry axis when you translate. And you have here 3 different options so that you can hit with a translation that goes always along a 5-fold axis, so we're going up. But depending on, you see the wiggle room here, going a bit more, a bit less, I hit one and not the others. So what I'm then therefore getting are different group relations. Because one, for instance, meets at the 5s. So I write those relations down that sort of meet at the 5. They will involve a 5-fold rotation most likely, and so on. The ones at the 2 will most likely involve a 2-fold rotation or a 3. Now there is obviously you need only 2 generators, so a 2 and a 3 give you a 5 and any combination thereof. So say you have the 2s and the 3s chosen, right? So then you can imagine the 5 is a combination of the 2s and the 3s. So you get a different combinations of 2s and 3s to write the relations for that one down, then the ones with just the 2 and just the 3, and this will be a mix. So that's the way to see it. So group relations have more combinations than one.
Okay, that's fantastic. My follow-up to that is, could this be a way of improving structural stability? Because if there is a symmetry group by which you have the near misses, that's a mode by which you can absorb, say, structural stress.
That's a super good question. So indeed, and even more. So what I haven't mentioned is that you get, depending on where your coinciding vertices are, once you have used the symmetry, full symmetry, you can count how many points you have. So how many words you have in your extended symmetry group. And that will vary depending on which you've chosen. And what we find is that the ones that have the minimal number are usually the most stable. So when we design, we want to go with the ones that make the minimal cardinality on that extension. It's an excellent question.